Hey, this is Mr. Wyas. This is a tutorial on 3.2 on chromosomes. So we start with bacterial chromosomes. What are bacterial chromosomes like? How many do they have and where are they? So here we see a classic bacterial shape down here. This could be something like E. coli. And inside we have two different forms of DNA. So one's over here, and this is the chromosome. Now we could just draw it as a large loop. You can see they've put extra kind of overlaps in there, but if we were drawing it, we could just draw it as a large loop. And it's a circular chromosome. So bacteria have circular chromosomes, where most of their genes are, and then they can also have something else called a plasmid. And so this is like an extra piece. And here there are multiple, often it might just be one plasmid, and this extra piece of DNA has some special features. So they have a single circular chromosome, and they have plasmid DNA. So let's zoom in on this bacteria here and have a look at where the DNA is located. So as a quick review, these bacteria have lots of different things to label. So you can stop and see if you can label those things. What were those features that we looked at in ultrastructure in 1.2? They've got ribosomes, they have a cell wall, and then inside the cytoplasm, because this bit here is the cytoplasm, inside the cytoplasm there is this, and so we drew it as more of a circle there, here it's more wiggly, but they've just got a circle, a circular chromosome. And so this is in the cytoplasm, but sometimes we can call it the nucleoid region. So it's the nucleoid region, and that's where they have their DNA. And then over here we can see we've got the plasmid, another small, and usually they're drawn more circular, another small circle of DNA. So here's our summary. Prokaryotes, bacteria, have one chromosome, only ever one chromosome, and it's circular DNA, and therefore if they've only got one chromosome they can only have one copy of every gene. It's located in the cytoplasm, we could say the nucleoid, and if we zoom into it, it's naked. So the DNA double helix, which is going round in a circle, it goes all the way around in a circle, it's not associated with histone proteins. So we saw in eukaryotes we get this winding of DNA around histone proteins, but in prokaryotes they don't have that. So it's not wound around. And it also isn't in a nucleus, so we say it's not membrane bound. Not in a nucleus, doesn't have histone proteins, it's just in one continuous loop and they only have one. What is a plasmid then? Well, we said that they had some special features. Well, a plasmid is this small circle of DNA. And over here, we can see that this one here, this gene over here, in this plasmid, is for tetracycline resistance. So this is a gene which gives antibiotic resistance. And so these plasmids can be shared with other bacteria. They can extend out, and if we remember that they have these extensions, these things called pili, and they can reach out, and down the pili they can send the plasmid, and they can connect to another bacteria, and now that gene can be transferred. And this is called conjugation. So it's their version of sex in terms of exchanging information. So these plasmids, they can be used in biotechnology. If we want to check that a cell has got the plasmid that we want, we can put in antibiotic resistance, and then when you grow it on an agar plate, you can put in, if I put tetracycline on this plate, the only bacteria which will grow, the only things which will grow on there, will be the ones which have this gene. So it can be a way of selecting. But mainly these are in wild bacteria, and they develop genes for resistance, and then they can quickly spread into other bacteria. So these genes for resistance to antibiotics can spread. So a plasmid is a small circle of DNA which may have genes for things like antibiotic resistance. Here's the summary. Plasmids are small extra DNA molecules, and they are circles, so we can draw them as small circles inside bacterial cells. They're found in many different prokaryotes. They're very rare, and really the IB says they're not present. We don't have many at all in eukaryotes, so they say not present in eukaryotes. They contain a small number of genes, and it could be for antibiotic resistance, and they can be transmitted from one bacteria to another, so they can spread in a population. So how do we know how long these bits of DNA are? And this is a bit about techniques in science. So what technique was used? What could have been made to make this image here? So this image it's a bacterial chromosome. And we've got to try and visualize DNA. And DNA is too small, it's on the nanometer level, so we can't see it. Even with the most powerful microscopes, it's very difficult to see and visualize. And you wouldn't be able to see the whole piece of it. So they used a technique, again, where they were tracking, they were using radioactivity. And they used radioactive thymidine. But what is thymidine versus thymine? So we've heard of thymine. Thymine, when we talk about T, we're only talking about the base. We're only talking about the nitrogenous base. So nitrogenous base is thymine. But when we add that nitrogenous base to a sugar, which is deoxyribose, then we get something called a nucleoside. And the nucleoside, they have slightly different names. So thymine adds to deoxyribose and we get thymidine. So what did they have? They had a kind of radioactive thymidine. And they put this into the mixture that the bacteria were growing in. So if the thymidine that's going to go into the DNA structure is radioactive, it's going to give off radiation that can be detected. So these bacteria are growing in this medium with radioactive thymidine that they take into their cytoplasm, and they're using in DNA replication. So they're now incorporating this radioactivity. So if we think that it's giving off radioactivity, anywhere where we've got DNA, we can track that. So what they did was they left the bacteria, and it was next to a film which would develop. So the radioactivity would cause the development of the film. And we can see then in those regions where the bacterial chromosomes were. And then they could measure those distances. In order to get out the DNA, they had to break open the cell. And a fancy word for break is lysed. So they broke open the cells to get the DNA out. They put it next to a photographic film, and then they left it, and they had to leave it for, the period was 2 months for it to develop. It wasn't like quick photography film. And so over a period of 2 months, the radioactive thymidine gave out radiation which developed the film, and then they produced this. And this technique is called autoradiography. So autoradiography. It's the idea of radioactivity and kind of photography together. So here's our summary. So it was a person called Cairns. They used autoradiography. They grew bacteria, and it was E. coli, with radioactively labelled thymidine. And therefore, when the DNA was being copied, replicated, it would have this incorporated in its DNA. So they then broke open the cells, they lysed them, and then they took the lysed cell contents and put it on these photographic films, and then the radioactive DNA would create these dark marks on the film, and that allowed them to then measure the length of the DNA along there, and then they could see how long the DNA was. And this allowed them to work out the length of a chromosome in a bacteria. What about eukaryotes then? So we've looked at bacteria that have a single circular chromosome and sometimes plasmid DNA, but in eukaryotes it's very different. So in our cells we have DNA in a different format. So see if you can label this. What is this? What are the different features of it? Well, this is a chromosome, and in eukaryotes it's made out of DNA and protein. It's chromatin. They are linear, and so it's just a single line. They also come in pairs. So we don't have one, we don't have single chromosomes. That's not what we find in eukaryotes. They come in pairs, and at specific locations, the gene locus, we have specific genes on different chromosomes. They're associated with histone proteins, so it's chromatin in eukaryotes, whereas it's naked DNA in bacteria. Because they come in pairs, at different locations we could have different forms of the gene. We can have different alleles in the same individual. Whereas in bacteria don't have alleles because they don't have 2 copies of each gene. So our quick summary for eukaryotic, they're linear, straight DNA molecules associated with histone proteins. We have 2 copies of each chromosome. And so in humans we have 23 with 2 copies, giving a total of 46. And it varies depending on the species. Different species have different chromosome numbers. Where are they found? They're found in the nucleus, so they are membrane bound. Between each chromosome there are also differences. So what are the differences between the different chromosomes which we give numbers to? So here's 2 different human ones. What differences can you see between chromosome number 3 and chromosome number 22? Just stop and have a think. Well, the most obvious difference is they have very different lengths. So we have different lengths of chromosomes. We have different numbers of genes because they have different lengths. We also have this position here, the midpoint, the centromere. The centromeres are in different positions. And we can think about how long these are. These can be called arms. So the arms of the chromosome are different. And you can see here, this is a list of all the genes that we know are associated with different diseases. So you've got things like, if you can just about read, colon cancer, a gene there associated with colon cancer. Whereas over here, we've got other diseases like deafness. So So there's deafness over there. So there are different genes which are involved in different things, and they could lead to, if there are faults with them, different diseases. But always when we go to chromosome number 22, the genes will be in the same place for different people in the species. So on my chromosome number 3, there will be the same genes in the same location, the same locus. Here's our summary. So the chromosomes in eukaryotes differ in length, the position of the centromere. They might have different versions of the gene, so different alleles on each of the 2 chromosomes. But each gene always occupies a specific location on that linear chromosome called a locus. What are homologous chromosomes, and how are chromosomes the same? How are they different? So here we have a pair of chromosomes, and we brought them together. This could be, for instance, like we saw above, chromosome number 3. So we can see that these have the same length. They came from, one was from the mother of this person, one was from the father. So that's where they came from initially. Now we can see that they share a lot of things, and they share the same length, they're the same size, we could say. They have the same banding pattern. So if we stain them, they will have the same banding pattern. They all have the same centromere position because they are the same chromosome with the same genes at exactly the same point. So what is different about them? Well, even if they have the same gene and it does the same thing, these two could be different forms. So we have here slightly different colour representation. These could be different alleles, different forms of those genes. Here's our summary. So homologous, one thing to point out about homologous, homo means the same, and logos means structure. So they are the same structure. Now if they are the same structure, that means they are the same length. And you can only have the same length if you have the same genes, because otherwise you wouldn't have the same number of base pairs of DNA. So if they're homologous structures, and if they're homologous chromosomes, it means that they have the same length, the same shape, the same genes at the same gene locations. But we might have different alleles, different forms of them. So an example of this would be one person could have one working copy of the gene for hemoglobin. So one could be the normal copy, but at exactly the same location, they could have the faulty copy. So it's the gene for the same thing, and this was the gene for hemoglobin, but one of them could be working and one of them might not be. The other thing that they do that we'll see when we look at cell division is that they will form pairs during cell division. They can pair up, find their partner, and they also do this pairing up in making of sex cells that we'll look at in reproduction. Take a couple of minutes and have a look at the table there and think about what it shows about the genome size, the total amount of DNA in different species. So the IB included this. I think you're unlikely to be asked specifics about it. I think maybe you might be asked to give some kind of ranking in terms of what might have a bigger genome size, or to make some kind of comment that more complex organisms have more DNA and a larger genome size. But we can see here for phage, this is a virus. Viruses have very small genomes in comparison. So 0.18 million base pairs, so that's about 180,000 base pairs of DNA. Whereas E. coli has 5 million. And then we've got a human which has got 3,000 million, that's 3 billion base pairs. And so we mentioned this before, that we get plants that have more, and this is due to duplicated chromosomes and DNA. So it's not necessarily that if you keep on adding more and more DNA that you get more complex. Okay, if we consider what's inside a sperm and an egg, what DNA is in there? And how can we think about this? Well, these are both sex cells. And so if we're thinking about sexual reproduction, we are talking about two halves coming together to make the full copy. So inside is half the DNA required, and it depends on the species. And this half of the genetic information, so if we were talking about humans, we would have to have, and I'm not going to draw them all, we would have to have 23 of our linear chromosomes in each one. And the same would be in the egg. We'd have 23 and 23. So we have a term for this, and we say that they are haploid. Haploid meaning half of the genetic information. So when they come together and fuse to make the zygote, and we get back up to 46 chromosomes, this is called diploid or diploid. Di we can think of as meaning two, and this means that for every chromosome, so let's just make this one red, we would have 2 copies of each chromosome. Maybe that's chromosome number 1. And so in the diploid cell, we will have 2 copies of number 1, where one of them has come from the father and one of them has come from the mother. So here's our summary. A haploid nuclei contains 1 copy only of each chromosome. So it's half the normal copies. Formed during meiosis, which is the process of producing sex cells, and they fuse together during sexual reproduction, forming diploid cells. We said the first cell there is the zygote. It forms this diploid zygote, which in us has 46 chromosomes, or 23 pairs of chromosomes. And that means that a diploid nuclei, what is a diploid nuclei? It's nuclei with two full sets of chromosomes. So a set of chromosomes, if we considered a set of 3 chromosomes, then one set of 3 has come from the sperm, one set of 3 has come from the egg, and then when we join it together, we would have our full set, which is 2 copies of each chromosome. And so now this cell is diploid because it's got 2 sets of each chromosome, and that means because these have the same genes at the same locations, it means we have 2 copies of every gene. And these could be 2 different alleles, or it could be that you have the same alleles from both your mother and your father. But it could be that you've got some which are dominant and some which are recessive. So this could be, on this chromosome here, there could be a big T, on this one there could be a little t. If we're thinking of big T, meaning, so if it was in pea plants, that would be big T would give tall plants, and small t would give short plants. So when they come together, we could have 2 different copies of the gene, two different alleles, so big T, small t. In the case of a pea plant, because big T is dominant, this would be a tall pea plant. And one of the significant features of this is if one of these was a disease gene, so if small t led to a disease, then because this organism has a big T, for many diseases, if you have one faulty and it's recessive, and most diseases are, then you can still function correctly. So we can have many faulty genes and we can still function because we have another copy of it. So what about chromosome numbers in different species? Have a look at these. This is chromosome number 2 in humans, and we can see chromosome number 2. And in the other great apes, it's slightly different. And so all the evidence seems to be pointing towards the idea of a fusion event, that these two chromosomes combined and gave humans our 46 chromosomes when all these other species have 48. So why can't we interbreed with the great apes? Because we have a different number of chromosomes. And so the evidence seems to point to some kind of mutation that led to a fusion of these two, and that reduced our chromosome number. And so now we have 46 chromosomes, whereas the other great apes have 48. And so that means that if a human and an ape try to interbreed, because of their different chromosome numbers, it doesn't work. It's a defining characteristic of species that they have the same chromosome number. And indeed, if we have mutations that lead to a difference in chromosome number, it would now mean that they are different species and that they can't interbreed. Here's our summary. So a chromosome number is a distinguishing feature of a species. Species have different numbers of chromosomes. And even if they do have the same number of chromosomes, they would have a different structure to those chromosomes. But a different number certainly means that species are unable to interbreed, even if they are genetically pretty close. And that's why chimpanzees and humans are unable to interbreed, even though the difference in chromosome number is only 1 in the haploid cell. So even if you do have the same number of chromosomes, they will have very different structural features. So you can find some species with the same number of chromosomes as humans. So here's a sable antelope. This also has 46 chromosomes. But the antelope's going to have different genes at different locations and different structures to the chromosomes, so there's no way that we could have interbreeding with humans, even if we have the same number of chromosomes. And here's some things that the IB's chucked in. No idea how they'd ask you a question on this, but they've picked out some different species. Homo sapiens, we've got 46. Which we've got 23 pairs of chromosomes. And so you can see that a chimpanzee, we mentioned, it's got one more pair of chromosomes, probably because of the fusion event that happened to create Homo sapiens. And then we've got dogs that have more chromosomes. So again, the number of chromosomes, just because we increase it, it doesn't mean that we get a more complex organism. There are organisms with 1,400 plus chromosomes, and they are not more complex. But we see here with this kind of fairly simple organism, a horse threadworm, which has a far smaller genome, it does have a lower number of chromosomes. How is sex determined in humans? What are autosomes? How are male characteristics brought about, and what is this image down here? What's a karyogram and what does it show? And how can we use it in determining Down syndrome? So we're going to deal with all of this in one go. Have a look at this. This is a karyogram. And it's made through the process of karyotyping. And karyotyping is taking an image of all the chromosomes. And we would only see these chromosomes during cell division. So during cell division, they take a photo down a microscope and they arrange all the chromosomes together. So we'd never see them just like this underneath a microscope. In the old days, they used to take a photo and cut out by hand and then match the chromosomes. And so they're matching them by the length of the chromosomes. Because we saw that the length, the number of base pairs of a chromosome, is a defining feature of a chromosome. So they arrange them all. Just stop there and see if you can work out what is the sex. Which chromosomes are determining the sex? Well, down here we see that we have an X and a Y. And so it's the 23rd pair which determines sex. And that means that the vast majority, everything else, all these other chromosomes, are not determining sex. And so if they're not determining sex, these are called autosomes. So autosomes are the non-sex chromosomes. And we could say they were autosomal. So many things, almost everything, is autosomal. Because almost every gene is on the chromosomes that are not the sex chromosomes. And then some things are sex determined. And so that can apply to diseases as well. So many diseases are called autosomal diseases, and very few are sex-linked or sex-determined diseases. As we can see that this person has one X and a Y chromosome, the Y is what gives maleness. It leads to male characteristics. So this person will be genetically a male. And if the person had had two of the longer chromosomes, two of the X chromosomes, then they would be female. In terms of the chances of you being male or female, at the top, if we have the mother who can make sex cells, which have half the copies of all chromosomes, that includes one copy of the sex chromosomes. If you're a female, you have two of the long X chromosomes. And that means that if you make an egg cell, then into that egg cell, the only thing you can put is the long X chromosome. And so a female does not determine sex. Whereas if we're talking about the male, the father could put into the sperm the long X chromosome, or could put into the sperm the short Y chromosome, which has genes for male characteristics. And so if we look at the odds of getting offspring, well, if the male puts in an X chromosome, then we get two long X chromosomes, we get XX, and you get a baby girl. And so that's going to happen half the time. And then the other half the time, the male is putting in the shorter Y chromosome, and now we are getting an XY, a boy. And so this makes you realise how silly it is that in some cultures women are blamed for not giving sons to males, but actually it's the males determine the sex. So why does the Y lead to maleness? Well, the Y chromosome has very few genes on it. It's only got 200 genes, and they're only known to make about 72 proteins. which is a very low number compared to other chromosomes. But it's got one gene which is very important. It's got something called SRY, and this is also called TDF. It leads to a protein that's called TDF, and SRY is the gene, and it switches on maleness. It switches on the development of male characteristics. So it's kind of a master switch. It's one gene that leads to a cascade of changes. And so this one gene SRY, if you do have SRY, then you create TDF. So if you've got a Y chromosome, you start making TDF. And so early on in embryonic development, when you are still developing in the womb, if you're male, you switch on TDF, and the TDF leads to development of your testes. And the testes then will start to release two things. One, MIF, it inhibits female genitalia being produced. And this bit here is a bit beyond the syllabus. So there's one inhibitory. The thing that we might be asked about is the testes cause the release of testosterone. And so as they release testosterone, we're going to get the stimulation. Testosterone now switches on the genes for developing male internal genitalia, and it also switches on through mechanisms that you don't really need to know, so these you don't really need to know. It switches on external male genitalia. So the flow is, if you've got SRY, you produce TDF, and TDF causes production, development of testes, leading to testosterone, which switches on the genes for male genitalia. Now, if you're a female, you don't produce any you don't have SRY, you don't produce TDF. No TDF means that you start producing ovaries. And because you're not making any of this inhibitory substance, you won't make any male structures. And so you've got no testosterone, and without testosterone, without SRY, without TDF, you'll develop female genitalia. And so this raises interesting questions about intersex individuals, because we have people who are XXY. And so sex, even on a biological level, is not a fixed thing. You could have a Y chromosome, but you can have issues with TDF switching on, or issues with testosterone being produced. And then that can lead to people who are actually genetically XY, being physically, in terms of their genitalia, they could have female genitalia, but they are XY or XXY. So here's the summary of all those points. So sex is determined by the sex chromosomes, the X and the Y. XX is female, XY is male. 50-50 chance, because sex is determined by the Y chromosome. With male characteristics being brought about by the SRY gene on the Y chromosome. The SRY causes the development of male characteristics through development of the testes and production of testosterone, which then switches on genes for male development. We can see sex and also other genetic diseases by looking at a karyogram. So karyotyping produces a karyogram that shows chromosomes arranged into homologous pairs, arranged in decreasing length. So we start with the longest chromosome and we go down to, at the end, the shortest chromosome. So the only exception to that is that the X chromosome is quite long, but it's still considered, the sex chromosomes are considered pair 23. So a karyotype can also show the the length of the chromosome and the position of the centromere. So the final question is, how can we look at a karyogram and see someone who has Down syndrome? Well, we zoom in on chromosome 21. And here we can see 2 copies. This person is diploid for chromosome 21. They've got 2 copies. But if we saw another copy here, one extra chromosome 21, then this person would have Down syndrome. So Down syndrome, it's called a tri because they've got 3 chromosomes. It's a trisomy. And there are different trisomies that can happen. You can have 3 of chromosome number 18, for instance, which is a different disease. But trisomy means 3 copies. It's called trisomy 21, and that is Down syndrome. And it leads to, you might think having extra chromosomes would be better, but by having more of those chromosomes, you get an increase in production of some proteins, and this leads to the symptoms of Down syndrome, including mental retardation. So they do this using a sample taken from the amniotic fluid to check. It's called amniocentesis. You use a needle and you take fluid, and then you do a karyogram. You can see if there is an additional copy of chromosome 21, then it will cause Down syndrome, which is trisomy 21. Now the downside of this is that this can lead to the loss of the baby. It can lead to a miscarriage in 1 to 2% of patients. So many people you don't want to do it unless there are other signs, and they have these through blood tests. So blood tests give an indication. And that's it.